So you've heard of the dodo and the woolly mammoth, two famous extinct species that will never walk this earth again. But what about all of the potential future extinct species that we still have the power to help save today? Like the black-crested gibbon, threatened by deforestation, or the sunder pangolin, poached for its scales. In fact, did you know that more than 1 in every 4 mammal species are currently endangered? And to protect them, we need to know where they are and how and why their populations are changing. To do this, we use camera traps, devices tied to trees that automatically snap millions of photos of passing wildlife. But we have a huge problem. Images are being collected far faster than humans can label them, leaving them unused, stuck on hard drives. In short, gathering the raw data is easy. Understanding it in its abundance, that's the tricky part. Now, you might think that artificial intelligence already has this covered, but computer vision models, which process images, require vast amounts of labeled data during training, and they can only be deployed in the particular ecosystem that they were intended for. For example, a model designed to spot a deer in a sunny meadow will suddenly fail to recognize a deer in a snowy forest. Due to this, specialists are forced to hand label thousands of images to reteach models every time a new ecosystem needs to be assessed. As you can imagine, this results in a significant bottleneck in the pipeline. So what can we do? That's where my research comes in, aiming to circumvent this issue by applying the principle that birds of a feather flock together. What do I mean by that? Well, machine learning algorithms are notoriously good at noticing patterns in data, and they can be taught to understand shapes, like the curve of a tail, or textures, like fuzzy fur, so well that they can group together similar-looking images without actually knowing what it is they're looking at. They're essentially providing mathematical fingerprints for each of the images, which can then be compared. But for simplicity, let's instead consider this jar of sweets, where each individual sweet is the image of an animal, and the colour is representative of its species. Now, all of the greens look pretty similar, and all of the oranges are quite alike. So via visual similarity alone, we can organise all of the sweets. Now, if I take one green sweet, taste it, and find that it's lime-flavoured, it's likely that all of the other green sweets are lime-flavoured too. This means that a labeller only actually has to identify a small handful of example points from each cluster or flock in order for all of the others to be automatically labelled as well. And the best part is, my research shows that having only 20% of the manual labels is sufficient for my method to achieve comparable results to training via the traditional approach. That's 5 times less tedious labour for the same outcome. So by rescuing lost data and by making models adaptable, I'm providing the tools required to scale up biodiversity monitoring to a truly global level, and fast, because time is of the essence. How quickly my models can spot species like these out in the wild might determine whether we humans ever see them again. Thank you.